Hello students, in today's presentation I am going to explain a topic from medical microbiology, that is candidiasis. Hope it will be easy for you to understand. Shall we start? In candidiasis, I am first going to tell you about the causative agent of the disease, its morphological and cultural characters, and then about the disease, the pathogenesis and clinical symptoms. Depending on the site of infection, there are different types of candidiasis. I am going to talk briefly about some of them. How to detect this disease in the laboratory, that is lab diagnosis. The prevalence of the disease in the community, that is epidemiology. How to protect ourselves from this disease, that is prophylaxis. And also how to treat the infection. Candidiasis is the fungal infection of the skin, mucosa, and rarely of the internal organs caused by the fungus Candida species. Out of more than 200 species of this fungus, the most commonly encountered are Candida albicans, which causes about 90% of the infection in human beings, and other species are Candida glabrata, Candida bullermondi, Candida tropicalis, Candida pseudotropicalis, Candida krusei, Candida lusitaniae, and Candida parapsilosis. Now, coming to the main species that causes this infection, that is Candida albicans, it is an opportunistic pathogen. What is meant by opportunistic pathogen? See, this organism is a normal inhabitant of human skin and mucosa. In fact, it lives as harmless commensal in our gastrointestinal and genitourinary tract. It is found in over 70% of the population without causing any disease. But when there is a favorable condition for it to overgrow, it will cause the infection, which in turn will lead to the disease. Before talking about the morphology of this pathogen, let me first refresh your memory on the morphology of the fungi in general. You know, fungi can exist in two forms. One is the yeast form, that is unicellular form, and the other one is the mold form, that is multicellular filamentous form. Some fungi are yeasts, whereas some fungi are molds. If you see the morphology of Candida, they are yeast-like fungus. That means they can either exist as unicellular forms or hyphal forms. In unicellular forms, they are oval or spherical shaped diploid fungus having a size of around 4 to 6 micrometer, and they reproduce by budding. Whereas in hyphal form, they can either form pseudohyphae or true hyphae. Pseudohyphae is a chain of elongated budding cells joined end to end and looks like a hyphae, whereas a true hyphae will have parallel cross walls, and this hyphal form is more important for an infection. Several factors can cause the change in this morphology, such as pH differences, temperature changes, carbon dioxide level, starvation, and quorum sensing molecules. Now coming to the cultural characters, the common medium for any human fungal pathogen is Saborodes dextrose agar. Candida forms smooth creamy white colonies with a yeasty odor in this medium. The optimum temperature for growth is 37 degrees Celsius. What makes a commensal organism like Candida to cause infection in the host? See, these are some of the virulence factors that we can find in Candida albicans. There are substances called cell wall adhesins that help in binding of the organism to the host cell via hydrophobic interactions. This reduces the level of yeast clearance from the body. Candida albicans also secrete degradative enzymes like proteinases, aspartyl proteases, and phospholipases. There is also a process called phenotypic switching that takes place in Candida, and this switching alters its adherence properties, antigen expression, and also tissue affinity. It results in the adaptation of the yeast to the unfavorable conditions of the host. Students, this is the end of part 1. In part 2, I will be talking about the disease, types of candidiasis, epidemiology, prevention, and also treatment.